Welcome back to the playlist on nucleotide catabolism and purine catabolism. This video will also be in pyrimidine catabolism. And in this video, what we're going to do is we're going to look at the degradation pathway for this molecule right here. And the molecule is basically deoxyribose 1-phosphate. Okay, deoxyribose 1-phosphate, which by the way is shown right here. So this molecule right here, this is deoxyribose 1-phosphate. This was the product of purine nucleoside phosphorylase, assuming that we were doing that reaction with a deoxynucleoside. Okay, so what we ended up doing is we had essentially up here in this position, there was a purine up there, right? And then we used purine nucleoside phosphorylase, and we cleaved off the nitrogenous base, and then we replaced it with a phosphate. Okay, so we ended up getting deoxyribose 1-phosphate. Well, we can't really do much with deoxyribose 1-phosphate, so we've got to get it into a form that we can use. And that's the final form we're going to get it into, which is deoxyribose 5-phosphate. Okay, so right here, this sequence of events that's going to occur is basically what's going to happen when we have excess deoxyribose 1-phosphate. So this right here, all of this business, this is the catabolic pathway for deoxyribose 1-phosphate. Okay, so the reaction that we're going to look at in this particular video is this one that's shown right here. This is called phosphodeoxyribomutase. Okay, you may be more accustomed to hearing this enzyme referred to by its common name, which is phosphoglucomutase. Okay, so this same enzyme is able to catalyze the interconversion of glucose 1-phosphate with glucose 6-phosphate. And so that reaction becomes important when you're doing glycogen synthesis and glycogen degradation. Okay, and so forth. So this enzyme is also able to catalyze the mutation of deoxyribose 1-phosphate to deoxyribose 5-phosphate. And again, the other name that you'll see this referred to as is phosphodeoxyribomutase, but they're the same enzyme. Okay, once we generate deoxyribose 5-phosphate, we're going to react that with an enzyme called deoxyribose 5-phosphate aldolase. And we'll look at the mechanism of that in a separate video, and that's going to give us two products. Number one, it's going to give us glyceraldehyde 3-phosphate, which notice is 3 carbons. And then we're also going to get this molecule right here, which is acetaldehyde, and that's 2 carbons. So this aldolase reaction takes us from something that has 5 carbons and splits it into a 3-carbon fragment and a 2-carbon fragment. And then we can take that acetaldehyde and react it with a very special kind of aldehyde dehydrogenase, which will get us to this molecule right here, which is called acetyl S-CoA. And then that could potentially go into the TCA cycle. So this right here, this sequence of events is what's going to happen if we have extra deoxyribose 1-phosphate. So now that we've seen the whole sequence of events, let's actually look at the mechanism of this enzyme. Okay, so number one, we're starting off the mechanism with deoxyribose 1-phosphate. And again, mechanistically, it's going to be identical to phosphoglucomutase because it's the same enzyme. So it's the same reaction, just on a slightly different substrate. So I'll do the mechanistic steps in green. So the first step is a base is going to deprotonate the 5' hydroxyl group, and that's going to induce nucleophilic acyl substitution on a phosphorylated serine in the active site. Okay, so that's an important thing to understand. This is technically a serine, but it's a phosphorylated serine, a post-translational modification to the enzyme. So as long as this enzyme is active, the serine is going to be phosphorylated. Okay, so it won't be a serine with a proton, it'll be a serine with a phosphate, and that's how this enzyme exists at rest. Okay, so when you attack this phosphate, okay, you're going to get a nucleophilic phosphoryl substitution. And I'm of course neglecting the pentavalent intermediate, but suffice it to say what happens is you'll get loss of a leaving group, which in this case is deprotonated serine. Okay, and that's what you see right here. So this is just the serine anion that's here in the active site. And now what you have is you basically have 1,5 diphosphodeoxyribose. Okay, because now you have a phosphate on the 1 position, which is right here, the 1', and you also have a phosphate on the 5', so it's 1,5-diphosphodeoxyribose. And then in the next step, what's going to happen is we're going to re-abstract that phosphate, but this time we're going to abstract it from the 1' position. So the serine anion is going to do a nucleophilic phosphoryl substitution on the 1' phosphate. So again, I'm neglecting the pentavalent intermediate that you would see, or you can call it a trigonal bipyramidal intermediate, but the phosphoryl intermediate is going to collapse back down and in the process re-abstract the proton from this base. And so the ultimate product that you get after this enzymatic reaction is complete is not only the phosphorylated serine, but you're also going to get 5' phosphodeoxyribose. Or another way you'll see this referred to as is deoxyribose 5-phosphate. And then this molecule right here, deoxyribose 5-phosphate, as we have just shown, is going to react with deoxyribose 5-phosphate aldolase, which is going to cleave a carbon-carbon bond, therefore it's a desmolase, into a 3-carbon fragment glyceraldehyde 3-phosphate and a 2-carbon fragment acetaldehyde. Okay, the topic of deoxyribose 5-phosphate aldolase will be the next video. But hopefully this by itself gave you a little bit of intuition on what's going to happen to deoxyribose 1-phosphate. In the next video, we'll look at deoxyribose 5-phosphate aldolase. See you in the next video.